Okay, hello everybody. We're here for lecture 4. We're going to be talking about uh, whatever this says up here in primary structure stuff. So you probably should have an idea of how the amino acids work at this point. Hopefully you have them all memorized. They're not super hard to memorize. I think the hardest thing is probably the pKa's and the one-letter codes because it can be sometimes be kind of weird. But other than that, let's get started. So don't really care, honestly. I mean, this is the thing. If you want to, yeah, I'll try and go over things that are actually important. So, we talked about this before. You need to look at the 20 common amino acids. So anything with like selenium in it or anything like that is not important. Essentially, mass amino acids are things that are obtained from diet, right? Yeah, proteins are made of L amino acids is important. You can't really use the RNS notation system. If you want to look at back at, like, I did an in-depth thing on this in the previous lecture for lecture 3, right? They're all made of L amino acids. The whole RS thing doesn't really work. Most of them are S, but like there's examples like cysteine, which is R, right? So try and rotate this molecule here in your head, and you'll kind of understand like how we kind of label things, right? And remember, when we do this, we're always looking at it from the perspective of like a Fisher projection, right? So the D's are coming out in dashes, right? They're coming out, right? So like wedges. Oh, sorry, those are wedges technically, but like the R group is out here on a dash. The COO minus is out on a dash as well. The H is out on a wedge, and the CH3 and plus is out on a wedge as well. And if you rotate this, right, you can rotate it in such a way where like the N is pointing, like if you rotated this like this way, right, like this R group would come out like towards you on a wedge basically, and then this would go away from you on a dash, right, or something like that. You can rotate it in different ways, right, to make it look a certain way. You want to try and make sure that this group and this group are in plane, right? So that would actually make this. That would actually mean you have to rotate this whole thing like this way, right? And so this R group would actually be on a dash, right? If you wanted this thing to be downwards. If you rotated it this way instead, right, this would actually be on a wedge. So just try and play with that in your mind a little bit. I talked about it in depth in the previous video, so if you need to look at that, feel free. These are things as well, right? There's different types of amino acids. Some are acidic, some are basic, some of them are donors, acceptors, all that stuff. So just keep that in mind. That's important to keep remember for just in general, right? So just keep that in mind. Here are all the amino acids. Again, hopefully you have these memorized by now. It's not too hard to memorize them. All the ones that have like amine side chains, right, are basic. All the ones that have this like carboxylate group are acidic, right? Anything that has like a hydrogen, like an OH thingy on it, or just an O minus or something, right, is a donor / acceptor, right? These are all acceptors, right? Basically anything that can hydrogen bond, things you've seen in bio already, right? Special cases are things like this thing. You'll realize soon enough that this actually can disulfide bond, which is very special for cysteine. You're never going to see this pretty much ever. You might get a question on it like in class, but it's like a hypothetical usually. They're not going to test you specifically on selenocysteine, but they could give you a question where they like give you a weird scenario, right, about selenocysteine, but they won't test you directly on selenocysteine. You got glycine. Glycine's a cool one, and proline's a cool one. These are both interesting because glycine's very flexible. You see it in like those disordered loops of proteins, right? And then proline is interesting because it causes forcible chinks in amino acid chains. So you actually can't put it in things like alpha helices and stuff because it actually messes with them. But yeah. All of the ones that are aliphatic or like hydrophobic are very obviously hydrophobic, right? The side chains just don't have any polarity to them. And you might look at like this and go, well, man, there's polarity to that. But remember the ratio of carbons to oxygens in this, and just like the ratio of carbons to like polar molecules in general, or polar atoms in general, is not very high. Remember that this N3H3 and O minus are actually part of the amino acid chain, the primary structure. So they're actually not positively or negatively charged. They're actually neutral in a protein, right? So it's just going to be this OH plus this ring, right? So that's quite hydrophobic. And this is obviously hydrophobic. It's mostly hydrophobic rings. I think the important ones to keep in mind here in terms of like interactions, I would say are, especially for those of you who are like interested in like MCAT. I think phosphorylation is something that they test a lot on the MCAT. Even in this class, phosphorylation is talked about a decent amount. So anything with a OH on it is phosphorylatable, right? So this is phosphorylatable, this is phosphorylatable, and tyrosine is also phosphorylatable. And then there's also things called phosphorylation mimics, which basically mimic the phosphate group, and that would be aspartate and glutamate, because they both have this minus charge, which is what phosphate has, right? So if you ever get a question about like a phosphate mimic, or like if you were to replace like this amino acid so it is constitutively active because it's always phosphorylated when it's active, you say, okay, well, phosphates have negative charges, aspartate and glutamate have negative charges, probably the most likely candidate there, right? If you've probably heard of like receptor tyrosine kinases, like RTKs, that's what these come from, right? But other than that, I don't think there's really much else on this screen that's important. For this, these are just numbers you should memorize. The pK of the N term is about 9, right? And the pK of the C term is about 2. And the R group for any given amino acid changes, right? So glycine has no R group, so there's nothing there. Arginine is 12.1. Glycine is like 9.6, 10.7, something like that, right? I think it's actually 10.7 technically. Aspartate is like 3.6. Glutamate's like 4.2. These are things you just have to memorize, right? So just get that in your head. Yeah, again, these are titration curves, right? So like for this one right here, this is actually like this is glycine, right, as it says up here. But I want you to think about what this would look like if you had a third pKa. So how would this graph look if you had a third pKa? Like if you had another R group, right, that had another pKa on it. Think about that. Yeah, and then obviously Zwitter ion stuff. I taught you guys in the workshop how you can. I believe I talked about this in the workshop, how you can actually find the Zwitter ion, right? It's just a place where it's neutrally charged, right? And you can see here, it doesn't have to be that the molecule is like neutral and everything's neutrally charged. It just has to be that the charges cancel out and they're neutral. So keep that in mind, right? This is a resource for you if you want to do this, right? I would just say you should be able to name all the amino acids here, right? And it should be really fast, right? Tryptophan, isoleucine, right? Oh, sorry. No, sorry. I made a mistake. That is not isoleucine. Look at me forgetting all of my things now. Yeah, that's crazy. But yeah, this is tryptophan here. And I'm gonna have to actually go back up here. How have I forgotten these amino acids? Look at me. It's crazy, man. This is leucine. I always mix up leucine and valine when I was studying these, but I haven't had to use these for a while. Oh, I did have isoleucine, correct. Look at that. Okay, cool. Yeah, so tryptophan, right? Isoleucine, leucine, right? I feel like these amino acids honestly don't show up that much, but here they are. Right? Okay. Yeah, so tetrapeptide, cool. Nothing crazy here. Amide bonds, right? The amide bond is right here, right? When they talk about residue, right? Like amino acid residues, right? Residue is just like what's left over from the amino acid after it's been added to a chain, right? So that is the residue of isoleucine, right? Because normally, if you remember, like if you just look at lone amino acids, this is not connected to anything, and this is also not connected to anything, right? So, directionality, we always write amino acids from the N to the C terminus, N terminus, C terminus. So if the test asks you to write the amino acid sequence, make sure you're starting from the N terminus and not the C terminus, right? It doesn't matter if it's written right to left or left to right. Always start from the N terminus wherever you see this empty NH2 plus, right? Or the opposite end of this. You should never start writing at this unless the question specifically asks you to start writing it from the C terminus, which is weird. They would never ask you to do that. Yeah. 50 log pH peptide Zwitter ion. I actually don't know. I mean, I guess you can do the number line technique here, right? So I mean, this is around 9. This is 2. There's no other things here that have pKa's, right? So you just have 2, 9, and it would just, yeah. I mean, Zwitter ion would be in here, right? So physiological pH is around 7.4. That would be like here somewhere, right? That makes sense, right? You need to draw out the whole thing, right? If it's below this, it'd be +1, 0, -1, right? So you can tell that the Zwitter ion would be in this range here, right? Here's peptide backbone, obviously, right? You should be able to identify this quite easily, right? But obviously, you might need to practice. Gosh, why am I coughing so much? Jeez. Whew. But yeah, you should be able to identify this, right? Start from the N terminus, right? Find the C terminus at the end, right? And then just kind of follow this snaking path here, right? And then everything else that comes off of that is a side chain, right? And you can also easily tell based off of like following these like carbonyl groups, right? Follow the carbonyl groups. You follow the amide bonds, follow these amide bonds here, right? You should see this like this structure basically a lot, right? It should be a repeating pattern of this, right? It should alternate from being up, the carbonyl being up and down, but that's not always the case. Sometimes they write it in different ways, right? Yeah, here we go. Here's the codes for these, right? You should be able to recognize these now, right? If not, make sure you memorize these, right? These are very important. Yeah, it's just numbering the R groups, right? Nothing crazy. Again, if you've done any sort of practice or have any sort of idea of the amino acids, this is pretty dang straightforward. This is usually the first question on any given 440 test for the first exam is usually just like a peptide thing of just identifying peptide stuff. So if you get this down, it's easy free points, honestly. Yeah, so this is actually an interesting one. You should be able to actually tell me the peptide sequence for this, right? And it shouldn't be, you should be able to do that relatively quickly, but obviously no pressure here, but I'll give you a second to think about that. Okay, let's go ahead and take a look at this, right? So you start from the N terminus, right? So here, N, right? And you don't actually have to write this usually when you actually write down on a test, but for the sake of being clear here, I just want to make this very obvious. This is the N terminus, right? So this right here is a cysteine, right? I can tell because this is actually a disulfide bond, right? Also, the R group is supposed to be like on this carbon, right? And I can see that this is the carbonyl group here for that amino acid. So this must be the side chain for that amino acid. So this is cysteine, which is C. If I look here, this is the next amino acid, right? Because this is the amine from the previous amino acid. This is supposed to be tyrosine, but what the heck is going on here? That's not tyrosine. That's tyrosine's fake cousin, right? So this is some form of tyrosine, right? Tyrosine is Y, right? Actually, you know what? Let me go ahead and I'll just go ahead and type these out. Probably a bit cleaner, right? Right, and okay, the next one here is isoleucine, right? So this is isoleucine right here. You can tell by this side chain here, right? So I'll go ahead and say isoleucine, which I believe is I, if I'm not mistaken. This right here is what is this here? So this is is either asparagine or glutamine, right? So usually if it was going to be asparagine, there'd have to be the carbonyl here followed by the amine, but it's not the case. So it's one extra carbon, so it's actually glutamine, and glutamine is actually Q. It's one of the weird ones. This right here is asparagine, right? This next one literally right next to it is asparagine. This is asparagine right here, right? So that would be N. One of the other funky ones, this is proline, right? Oh, no, it's not. Wait a minute. Hold on a second. I missed one. This right here, right? I missed this one right here, right? This is another cysteine, right? Because it forms another disulfide. This forms a disulfide bond. The one following it is proline, right? This weird guy here is proline, right? And then the one following that is a leucine, right? So just keep that in mind. That should be your peptide chain, right? You should be able to label this, right, pretty easily. But it took me a bit there to get that right. I was talking through it, but on an exam, I'd probably go through that a little bit quicker, right? If I was in my prime of 440 knowledge. Okay. You've probably already seen these from BIOC 200, honestly. The primary structure is just a sequence of amino acids, so like this is an example of a primary structure, right? It doesn't really have any sort of secondary like structure to it. And here we go. Here's an example, right? Primary structure right here. The secondary structure is where you get like the alpha helices and beta sheets, right? So that's something to keep in mind. So if they ask you about secondary structure, just think alpha helices, beta sheets, disordered turns, all that stuff. Tertiary structure is just like multiple combinations of alpha helices and beta sheets, right? So this is an alpha helix, right? You can see there's an alpha helix here, there's an alpha helix here, alpha helix here. So basically, you just combine a bunch of them, right? And then a quaternary structure is just a combination of multiple tertiary structures, right? So you can see here's one tertiary structure, here's another, here's another, another. So if you, you'll learn later, but I believe pyruvate dehydrogenase, that entire enzyme complex, is an example of a quaternary structure, if I'm not mistaken. So yeah, same thing with ATP synthase. I believe that is also multi-subunit quaternary structure. So, these sorts of quaternary structures are usually held together by like disulfide bonds. That's how you know it's an oligomer, right? Oligomer meaning multiple, like a complex of multiple proteins, right? So yeah, you'll learn about like heterodimers, homodimers. These are different types of oligomers, right? So homo meaning same, dimer meaning protein. So two of the same protein, or heterodimer, two different proteins combined to make one. There's many different kinds of these, right? So a heterotetramer, homotetramer, homotrimer, heterotrimer, all sorts of these things, right? And I believe you guys will get tested on like STS page gels. So that's also something to keep in mind. I'll probably go over that in my like exam review workshop. I don't know if they really cover it here, so. This is just a nice cool slide about like how there's many different amino acid sequences in nature. For 100 residue protein, there is too many possibilities. There's a lot, right? So, not really something too important here in my opinion, but it's there. Hydropathic plots is interesting. I don't know if we ever really talked about this. Even if we did when I took it, it wasn't very important. Just keep in mind that anything that's negative is hydrophilic, right? So here's an example. Yeah, so like this thing, right? These like regions above 0 are all like transmembrane domains for a protein, right? So like transmembrane, meaning inside the membrane, right? Crosses the membrane, right? These are all transmembrane. Anything below this is hydrophilic, meaning it's probably outside the membrane, right? And you can probably tell based off of what's here. So like, this probably be inside the membrane. This probably be inside the membrane. This probably inside the membrane. Inside the membrane. Inside the membrane. Probably not. You can tell like this section here is probably inside the membrane, right? And there's different things like this that you can use to figure out if a protein section is inside the membrane or not. Probably inside the membrane. Membrane, probably, right? So these probably inside the membrane here, right? But yeah, you can use like different techniques to figure out what's inside the membrane. But for this slide, I would just say just remember that for these sorts of plots, these guys here, just remember that minus equals hydrophilic, or negative equals hydrophilic, and positive equals hydrophobic. Right? I think just remember this, and it's probably all you need to know. I can't find a place to put this. Go ahead and make this smaller. Yeah, I'd remember this, honestly. Okay. Yeah, secondary structures are interesting. I think what you should know about this slide is this. Glycine and proline are not supposed to be in alpha helix conformations. How do I know that? Because they have a very high positive delta G. If you remember, positive delta G is not spontaneous, right? Which means that it's not going to be incorporated, right? It's not favorable to incorporate a glycine or a proline into an alpha helix conformation. And we talked about that, right? Glycine is very flexible. You don't want a very flexible amino acid in your alpha helices. It will disrupt the structure and actually won't work. Proline introduces forcible kinks in a chain, right? You don't want to incorporate a forced kink in a chain that's supposed to be a spiral, right? It doesn't work like that. So these are just things to keep in mind, right? I wouldn't say anything else, and this is really that important, honestly. Yeah, so this is something you should know. You can predict like whether or not something is a beta sheet or an alpha helix based off of like structure, right? So something to keep in mind, I'd say. Is there anything here that's like really helpful though? I guess what I would say is like, let's see if there's a good example of this here, honestly. Don't know if there is. Generally speaking, if you're looking at like alpha helices, like every 3 to 4 amino acids should be like hydrophobe, like either hydrophilic or hydrophobic. Basically the way that works is that like every 3 to 4 amino acids, there's like an alpha helix turn, which means that all of the amino acids of that type end up on one side. I'm trying to see if that actually shows up here. So this right here, the E is like hydrophilic. 1, 2, 3, right? This is also hydrophilic, right? 1, 2, 3. I mean, tyrosine may be hydrophilic. It's kind of a bit of a weird one. 1, 2, 3. Threonine is definitely hydrophilic, right? 1, 2, 3. But you see what I'm doing here? Like every 3 to 4, right? See, you can see this, right? Like every 3 to 4 amino acids is generally speaking a hydrophilic in this case. It could be the reverse, right? It could be that like every 3 to 4 amino acids is a hydrophobic, right? That's generally how I was able to figure out if it was an alpha helix or not, and it worked pretty dang well. So let's try it for this one down here, maybe. Let's see. Let's see here. Starting from this serine, maybe this serine, or maybe like this asparagine. 1, 2, yeah, I think like, yeah, I'll start with this asparagine here. 1, 2, 3, 4. Yeah, let's see this D here, right? 1, 2, 3. This lysine, right? 1, 2, 3, 4. 4 in this case. There's a lysine right here. 1, 2, 3. There's another serine right here. There's asparagine here too, right? But you can see how this is like relatively consistent, right? So I think it's just something that's a useful tool to keep in mind. Let's see, for beta sheets, let's see. Ooh. For beta sheets, I believe the thing I used to use was that generally it would alternate amino acids, but let me double check real quick for that. Yeah. Yeah, if I'm not mistaken, generally speaking, I think the easiest way, honestly, to tell if it's a beta sheet or not is that there generally should not be any kind of like glycine or proline in there, right? Um, yeah, I guess like, yeah, okay, here, yeah. So I think one of the ways that you recognize that it's a beta sheet is, generally speaking, I think it's usually like one type of not one, like one type of amino acid. So like, it's usually like, it should be like a string of amino acids that are a certain type. So like, for example, in this case, you can say that, trying to find a good example of this here. And you like, even these are not great examples. Like, generally speaking, they're usually of the same type, but like, spirit, glutamine, isoleucine, lysine, valine, glycine, serine, phenylalanine, proline. Yeah, see, like, this is unusual for a beta sheet. I feel like you probably usually don't carry these. I'm actually not too sure about the beta sheets. I might have to do a little bit more research on that. But I would say for the alpha helices, for sure, keep in mind this I plus 3, I plus 4 kind of rule. And then for the disordered bends, I would say like keep an eye out for like prolines and glycine. Glycine is definitely the more common that you'll see, because glycine is very flexible, right? And you can probably see that in like a lot of these. Like, for example, here there's a glycine, right? So see this glycine here, glycine, glycine, right? You can see that in these like little bends that are coming up, right? No glycine there. There's a glycine here, right? So you'll notice it, like here's another glycine, right? There's the glycine here, but that might be inside the beta sheet, or like right before entering the beta sheet. So it's something to keep in mind, right? That these things are there. I also look a bit more into beta sheets. I can't quite remember the exact pattern that I used to figure them out. But yeah, let me see if I can find another. me see if there's like another thing I can find here. Yeah, I can't quite find it. Yeah. I'll have to look into that a little bit more, but hopefully I can come back with an answer on that in like the workshop or something. So just keep that in mind that that is possible. That they could ask you about those realistically. Yeah. These are things, AlphaFold. I use Pymol when I was doing like stuff in the biochem lab, so these are the thing. You'll use these if you do biochem lab, or if you're in a lab currently. You'll use these programs, right? Yeah. There is a way of predicting whether two proteins like predicting function based off of similarity, right? So homology, right? Homologs are proteins that are just derived from the same common ancestor, right? And these we're looking for proteins that are similar to each other in terms of structure, right? Yeah, so we basically just align these sequences, right? Do all this jazz. I wouldn't say these are super important. I personally didn't have any tests on these, so but I guess this is something to keep in mind, probably the two proteins, right? I mean, I've never personally had questions about this, but it's possible they could, right? So. Yeah, so here's an example of comparing myoglobin, right? I personally don't think this is that important. I guess if your professor said that this is something to keep in mind for the exam, sure. But like, they have so many better things to ask you questions about. So, this is something I actually talk about in Biochem 442, which is really interesting that they're bringing it up here. BLAST is basically just a way of comparing sequences of amino acids to see if they're similar or they have homology, right? And these like dashes, generally speaking, are amino acids that have been kept the same across different species, right? And you can see that, right? This is all proline, right? Proline, glutamate, and arginine, right? You can see here also proline, arginine, and lysine, right? You can see these are all conserved, right? And then there's these double dot regions. This dot, I think the dot, if I remember correctly, means that it's like relatively conserved, but not always. And then like the double dot means that they don't really care. There's some conservation, but nothing crazy. The stars are like the big conservation, like they are always the same pretty much. So yeah, like here, invariant positions, right? Conservation changes, right? So, right? Yeah. And here is a nice little visual of how this works. These are really weird looking. I'm not gonna lie. These don't really make a lot of sense when you first look at them, I don't think, but I can hopefully explain it really easily. Coloration, if you want them. I don't actually know what green means. I think it just means polar, but that doesn't really make sense because glycine is not polar, so I have no idea what it's trying to say there. But I think the important thing is to recognize from this is just like the probability, right, effect. So the larger the letter is, right, first of all, the letter tells you what amino acid it is, right? And second of all, the larger the letter is, the higher the probability of getting that amino acid in that spot is, right? So you can see here that there's like a point, like an 80% chance of getting a valine here, and like a 20% chance of getting an isoleucine here, right? But and like here, like these three here, it's 100% chance you get any of these. You get a proline, an arginine, and a lysine in these positions of the chain, right? And then like the x-axis is the amino acid number, right? So like you can see here, this is 1, 2, 3, 4, right? So at position 4, it's guaranteed to be a proline. Position 5 is guaranteed to be an arginine. Position 6, it's guaranteed to be a lysine. At position 7, it's like 70% going to be a glutamine and 30% going to be a valine, right? And you can see what I'm doing here, right? Or like 80%, 80-30, or 80-20 or something, right? You can see that like it's basically telling you what the percentage chance is of getting an amino acid, like this amino acid at that point, right? So these are important to keep in mind. They could ask you about these. I've seen test questions about this, so keep this in mind, right? Mapping conservation, 3D. Yeah. This is a thing. This is, I guess, I don't think they'll really ever test you on this. These things tend to show up in the center because, like, these are where the active sites are at, and active sites tend to be pretty conserved. The surface of the protein doesn't really matter that much, right, for enzymatic activity, so they generally just keep the center more conserved just because that's where the enzyme actually does its job, right? Yeah, this is something to keep in mind, right? So if you see sections where the amino acid is like conserved like this, the chances that this section is actually quite important for function. So if you see a question that's like, which section of this amino acid chain is likely important for enzymatic function, and you get one of these like massive diagrams, look for the sections that have these big letters that take up 100% probability, right? Like, so like this PRK, this PER, this R, E, and K here, even this like EED here maybe, but like definitely this R, this E, and this K. So these like these two here and this, right? Those are definitely conserved, right? And they're conserved for a reason, right? The enzyme probably needs them to function properly, right? Yeah, but this is like this is just a verbiage thing, right? So motif is just a short section of protein that's important, right? Yeah, and this is something you usually have shared metal, like shared function. I want you to think about, right? What kind of amino acids would you use for metal binding? Right? Just think about that before you look down at the amino acid sequence. But like, what would you use for that? Right? There's certain amino acids that are definitely better. Like, for example, calcium. Like, what would you use to bind calcium? What amino acids are good for binding calcium? Right? I personally would say things like glutamate and aspartate. Right? They have a negative charge. Right? They hold like they can bind that positive charge of a calcium ion. Right? Maybe you can argue serine and things like that, but I would say definitely the better answer is glutamate and aspartate, right? Keep that in mind. What if I wanted to bind a chloride ion for some reason, right? Lysine, arginine, positively charged, histidine, right? These are things to keep in mind, right? Again, they never asked me about this personally, but like, these are just like the ability to predict whether or not like what the amino acid is, right? Like you're trying to predict the identity of a residue, right? And you're basically looking at the notation here, but basically anything that's like set in stone is like we know it, and then like anything that's in these brackets is like it can be anything but these amino acids, right? So for example, like here, it must start with an aspartate, right? But like this second amino acid can be anything except for a tryptophan, right? Same thing here, like this amino acid sequence can be anything except for aspartate, glutamine, asparagine, glutamine, serine, threonine, alanine, glycine, cysteine, right? Stuff like that. Just keep that in mind. Oh yeah, look at that. Didn't even see this, right? So calcium. What do we need? Acidic. What's acidic? D and E, right? Right? That makes sense, right? So yeah. This is I don't really think this is important. I'm not gonna lie. Speciation, cool, is a thing, right? As organisms diverge, proteins evolve, right? To gain different functions or maybe to retain function, right? Um, Gene duplication, right? They usually don't ask you about this stuff. It might be good to keep in the back of your mind, but again, they have so many other things to test you on. I wouldn't say this is like top priority. Um, yeah, right. Again, I think I had a quiz question on this, so this is something to keep in mind, right? So orthologs is like homologs, across different species, right? Um, keep this in mind. I think this did show up on one of my quiz questions, but it did not show up on the exam, right? But yeah, I mean like it should be relatively easy to kind of it's not easy to read this, but like you should be able to tell, right? Like more similar up here, less similar down here, right? Here's the different names for different like um different species, right? So these are just things to keep in mind, right? Like that basically the idea here is that like a lot of species actually have very similar proteins, right? Protein structures. So yeah. Yeah, I would keep this definitely. So, I'd keep orthologs and paralogs in mind. Um, so these could show up on like a quiz, but the probability that they show up on an exam is quite low. But it's good to keep in mind. Like, you never know, right? So yeah. Uh, yeah. I would say there's not really much else here. I'm trying to think if there's really anything else important for this slide here. Rise from gene duplication. Search picks up other globins. All my two points. Yeah, I would say there's not really that much that's important here, honestly. Same thing here. Sequence comparisons, evolutionary. Yeah. Again, this is just comparing sequences, right? We're looking for homology, right? How conserved is it? How conserved is it not? The little bars show you like how conserved it is, right? So the higher the bar is, right? So the higher the bar, the more conserved it is, right? Lower the bar, the lower conservation it is, right? So. Yeah. I honestly don't think a lot of this stuff is important, but like, again, keep it in mind, but I wouldn't stress over this stuff. Other than that, that's pretty much it for this lecture. Hopefully it's a little bit helpful. I know it kind of went quickly, but there's really not much to this lecture, other than like the structure of proteins, right? Again, we're looking at primary structure, right? And we're looking at structure of proteins. So if you don't know your amino acids, that's where this will become a struggle, right? But like, just memorize these, right? And you're pretty much golden, honestly. Memorize those. Understand how the pKs work, right? The Twitter ions, very important, right? Be able to name these, right? 3-letter code, 1-letter code, pKas, all that jazz, right? The structures, right? So the primary structure, secondary structure, tertiary, like these things, right? Keep this in mind. But like, these are not, this lecture in particular is not the most difficult one you guys will get, right? So just focus on getting the amino acids down, focus on understanding how these structures work, right? What is the primary structure? What is the secondary structure? What kind of structures make up secondary structures, right? What amino acids can be used in different secondary structures, right? Tertiary structure, right? Quaternary structure, just keep these in mind. Other than that, though, I wouldn't be too concerned about this lecture. Hopefully it was helpful, though. If it was, great. If it wasn't, cool. But yeah, have a good rest of your day. See you later.